Today's topic is plant tissue culture. What is plant tissue culture? It is the technique which is used for the regeneration of a new plant from the small tissue or small plant cells. Okay, and we should maintain the environment as well as the culture media which is responsible for the growth of that plant cell and plant tissue. So, definition of plant tissue culture, it is the technique of growing plant cell and tissues in the artificial prepared nutrient medium which is called as culture media under aseptic conditions. These medium may be static or liquid. The culture medium may be static or liquid. So the procedure, actual procedure, here we are taking one part of the plant, leaf, then we have to cut into small pieces, then we are embedded in the culture media which is in petri plate. This process is called as culturing. After some time we get callus formation. Callus is nothing but the undifferentiated mast cell. Again, we will transfer this callus into the fresh media, and this process is called as subculturing. Okay. Objectives of plant tissue culture. So first, propagation of large quantity of good quality. See, the quantity and quality will be greater than the previous plant tissue or plant cell. Okay, and this method is beneficial because it does not require so long time, so it will occur within short time. Then space is minimum, cost is also minimum. Okay, second is obtaining disease free plant. We can regenerate the disease free plant from the disease one, okay, by using this technique. Then rapid propagation of plants, which is difficult, so by using this technique we can easily done this process. Next is genetics improvement. So genetically improved plant we can regenerate. Production of multiples of plant in absence of seeds. If we do not have seed, then how we can multiply the plant? By using plant tissue culture technique. Okay. Next point is preparation of culture media. Culture media which is used for the growth and propagation of the plant tissue. So how we can prepare that culture media? See, first we have to prepare the concentrated stock solution of different macro and micronutrients or elements which is essential for the growth of the plant. Next we have to dilute it and mix together for the adjustment of pH. pH should be adjustable because it is important factor for the growth of the plant. Next is addition of gelling agent. Why gelling agent is necessary? For the solidification. See, this culture media is in liquid state. Then we are pouring into the petri plate. After pouring, this should be solid. Then only there is callus formation. So because of this we are adding gelling agent. Next is autoclaving or sterilization for kill the to kill the microorganisms, to eliminate the contamination. Next, pour this gel into the culture vessel or petri dishes. After pouring, it will get solidified. Okay. Next is preparation of explant, the part which we will use for the culture. So how we can prepare that part, or how we can prepare that explant? Explant which will be regenerated, that plant is called as explant. So first we will select the source of plant which should be disease free. Then we have to cut into small pieces. Then we have to sterilize it. Okay, after sterilization again we have to cut into small pieces and placed over the culture media. This process is called culturing, as we already discussed. And if after formation of callus we will replace the media with fresh culture media, That process is called as subculturing. Okay. Next point is general steps involved in plant tissue culture. There are some steps which involve in this technique. So first step is preparation of medium and sterilization in autoclave. So medium which we are using as a culture medium, it should contain micronutrient, macronutrient, then vitamin, iron, growth regulators which are required for the growth of the plant, and the medium should be sterilized. So how we can sterilize? By using electrically operated autoclave, and this sterilization is important to avoid the bacterial contamination of culture media. Okay. Next is selection of a mother plant and sterilization, that is preparation of explant, as we already discussed. But here, see, the mother plant which is selected, it should be virus free, it should be disease free, and it should be healthy, so we can get the healthy plant after regeneration. Okay. The actual part which is called as explant, we already discussed it, and it should be washed with water, liquid soap, and antiseptic solution. After this, we have to wash with chemical solution. To remove out the excess chemical solution, we will again wash with distilled water. Okay. Next is inoculation of explant on sterilized medium of non-composition. See, here the explant which is inoculated on the sterilized medium, the inoculation takes place in the inoculation room. There is special room for the inoculation, so it will take place only over there. Then there is presence of laminar airflow which maintains the continuous airflow. Okay. And which keeps the bench without the risk of contamination. Okay. Next is shifting of culture to the growth room. See, after inoculation room is for the inoculation, and growth room is after inoculated culture, it is shifted into the growth room, and there is temperature maintenance which is in between 15 to 20 degrees Celsius, and there is artificial lighting which is important for the growth of the plant. Next step is subculturing of cultured seed. As we already discussed, subculturing is what? See, callus cultures are formed. Then after that we have to replace this culture by fresh culture. This process is called as subculturing. Okay, and it consumes the provided nutrients over the time. So to maintain the growth of cell, fresh nutrients are required. The cells are transferred to the fresh culture media, and this process is called as subculturing. Okay.